Okay guys, so we are going to go ahead and look next at the difference between monohybrid and dihybrid crosses. But before that, I want to make sure that we can understand exactly a little bit more about this vocabulary. So, for example, when we talk about true breeding individuals means that you only ever produce that specific trait. So, for example, if you are true breeding purple flowers, what is the only type of flower you'll ever produce? Purple. If you're true breeding yellow seeds, what is the only color seeds you'll ever produce? Yellow. Again, true breeding just means you'll ever produce that specific trait. Okay? Go ahead and put your Chromebooks at closed or 45-degree angle because we're not using it. Okay? But what we're doing though is taking true breeding individuals that are in the P generation and we're cross-fertilizing. What does cross-fertilizing mean? It's essentially sexual reproduction where we're taking the male and female flowers and we're pollinating the female flowers with the male pollen. Does that make sense? For those of you that don't know, flowers actually contain both male and female parts, or sometimes flowers contain only male parts, and sometimes flowers only contain female parts. Okay? But for example, this is on page 12.2, that small Punnett square. Again, I know you guys can do a Punnett square, so I'm really not really too concerned about it, okay? But I think it was purple right here. So if purple is dominant, then what would the alleles look like? If the purple is dominant, what would the alleles look like?
Uppercase?
Just one?
No.
It would be capital, just like that, okay? If you have white flowers, what would the alleles look like?
Lowercase b.
Again, that's the genotype, right? So if this is the case, again, always start with the top. So again, these were true breeding purple flowers because they only ever produce what? These are true breeding white flowers because they only produce white. But when I crossbreed them, and this is, I'm producing the F1 generation right here, my F1 generation is going to be 100% what? Purple. But what about the genotype? 100% what? 100% heterozygous. Genotype would be 100% heterozygous. Phenotype would be 100% purple. Again, what did I do? I cross-fertilized what? True breeding individuals for purple and white flowers to create my F1 generation that is 100% purple, 100% heterozygous. Does that make sense? Again, I know you can do a Punnett square. I'm not too worried about it. I need you to understand the vocabulary behind the Punnett square, meaning dihybrid, monohybrid, true breeding, cross-fertilizing, all of those words. Okay. But where am I? Oh, when we do this though, this is what a monohybrid cross looks like because I only crossed one specific trait to get a hybrid, which is the heterozygous individual that I see here. Okay. But in this next one, I'm going to take these babies, these offspring, and I'm going to self-fertilize them. What does that mean? It's an asexual reproduction, which means I take the pollen from this flower and put it on the stigma of the same flower. Okay, self-fertilizing. So that means what? Again, I have heterozygous. This again, this is the second Punnett square on page 12.2. So again, top down, capital P, lowercase p, capital P, lowercase p, lowercase p. Now, what is my phenotype? Three to what? One. Or I could also say 75% what? What color would they be? Purple. And 25% what? White. My genotype would be one what? 1 to 2 to 1. Or 25% what? Homozygous dominant. 50% what? Heterozygous. Or 25% what? Homozygous recessive. That's it. Again, what did I just do? I just did a dihybrid cross. Again, or not a dihybrid, a monohybrid cross, but to produce the F2 generation. I'm still looking only at flower color, because monohybrid means you're only looking at one specific trait, okay? Again, to clarify, if you're just doing a Punnett square that's just random and you're just saying that the alleles for the mom was this and the alleles for dad was this, and you do a Punnett square, that's a Punnett square. A Punnett square is not the same thing as a monohybrid cross. A monohybrid cross is crossing in the P generation two true breeding individuals, again, crossbreeding, cross-fertilizing, two true breeding individuals and getting an F1 generation. Does that make sense? The cross, monohybrid cross and dihybrid cross, it's a series of very, very specific steps that have been through this part right here, okay? If you're just doing a random Punnett square, it's not the same thing. You're just doing a Punnett square, okay? Which you'll get the same results, but it's just a Punnett square. Okay, now we're gonna do a dihybrid cross. So it will be something very, very similar to this on the exam, but as you can see in this, so as you see here, we're not gonna do this one just because I think this one is very easy and straightforward. I'm gonna let you guys do this, and on your notes, when I go to check, I will look for this one. But you have yellow and round and green and wrinkle. Yellow and round are both dominant traits. So what is yellow and round going to look like? What would the genotype look like for these traits? If yellow and round is dominant. But what letter did I use? Uppercase Y. Why Y? Why not G? Because as you notice, whatever the dominant allele is, the phenotype is usually the letter that you go for. Okay, like purple being the capital P instead of Y. Okay, or W. So this would be like this. And then capital R for what? For round, and then green would be
Green.
lowercase g?
Lowercase.
But yellow is dominant. That means green is recessive, so what letters would I use? Lowercase y. Again, we use capital P for purple and lowercase p for what? White. Capital Y for yellow, lowercase y for what?
Green.
Green. That's what I mean. Okay. Capital R for around, lowercase r for what?
Wrinkle.
Wrinkle. So that's what they would look like. Okay. I'm gonna foil them, and then I'm gonna put this one on the top, this one on the side, do my whole dihybrid cross, get my result. And just by looking at this, just like we've seen with this one that we just did, we're gonna get 100% what? Again, these are the parents. They're true breeding for yellow and round, true breeding for green and wrinkle. They're gonna be 100% yellow and round, because yellow and round are dominant. So that means these are gonna be heterozygous, okay? But this is what I mean. They're gonna be yellow and what? Yellow and round, which is capital Y, lowercase y, capital R, lowercase r, okay? But now I'm gonna take my F1 generation and I'm going to self-fertilize to produce my what? F2, but this is what we're trying to see, okay? It's not gonna be the same thing. But we have to foil this to be able to properly put or set up this Punnett square. So how do we do a foil? Just like in math, we are going to go ahead and I'm gonna go ahead and show you. We have capital Y, lowercase y, capital R, lowercase r. Again, I don't know how comfortable you are with foiling, but I'm gonna show you how to do it, okay? First letter to the inside, so that's capital Y, what? Capital Y, capital R. First letter to the outside, capital Y, lowercase r. First letter to the what? Inside. First letter to the
House.
outside. Okay. And then I'm going to go ahead and do second letter, which is the lowercase y to what? Inside, which is a capital R. But Mr. Silva, I thought you couldn't put recessives first. Again, this is not the answer, we're just spoiling, okay, so that's fine, okay? Second letter, outside. Lowercase y, what? Lowercase r, okay? If you are unsure on how to FOIL, we're gonna practice, okay? I don't know, just, that's it. That's right. So, we are gonna go ahead and take this, yes. After this. Hurry, no, just actually just wait. 'Cause we have- Okay, so I'm gonna do a random one and I want you guys to FOIL that one. Again, this is just practice, okay? Capital L, lowercase t, lowercase t. Should FOIL it. Okay, go ahead and look at your sticky note. Don't look up until you are, want to see the answer, okay? Okay, again, when you're done, look up. And if this isn't what you got, you might need to see if you got it, okay? But if you did get it, great, love it. Let's move on. Okay, so the point of this is we are going to go ahead and do a dihybrid cross, which is the second one, which is again, that second one on the page 7.3, okay? Which should look like this. So, what are our genotypes? It should be capital Y, capital R, capital Y, capital R, capital Y, what?
Lowercase r.
Lowercase y, capital R, lowercase y, what?
Lowercase r.
Lowercase r. I'm gonna do the same for both sides because again, this is self-fertilization. I took my F1 generation and I'm self-fertilizing them, or in other words, it's asexual reproduction. I'm taking literally like a little paintbrush, taking the pollen, putting it on the stigma of the female part of the flower, and self-fertilizing it. It's exactly what Mendel did to make sure that his experiments were perfect, okay? Again, the reason why he gets credit for being the father of genetics, okay? One thing you'll see is that when I do my lowercase and capital letters, especially for those that look very similar, like Y's or S's or U's or V's, I always try to be very specific in how I write them. So for example, like the Y, I'm making the Y bigger, and the lowercase y, you see how small I'm writing it? I'm very, very specific. I make sure that I know and I can tell the difference, okay? Because when you're in the, like on an exam and you're rushing through it, you don't want to make mistakes, okay? Does that make sense? Okay. So, one of the biggest things that I always do is I always, especially with the hybrid crosses, always do top down. Do the top one, I'll go all the way down, then do the what? Left side, and go across. What I mean by that is this. I'm going to imagine that there are 4 slots each of these. And if you notice, I do not put them all next together like that. I just put them, these two and these two. What do you think is gonna go up here? The Y's or the R's?
Y,
And over here are gonna be the
R's.
R's, just like that, okay? Capital Y is always gonna go first because it's a capital letter, right? Capital R. Does that make sense? But since I did this first one, what is this one gonna look like?
Capital,
Again, I'm ignoring the left side, I'm only doing the top and bringing it down. Capital Y, capital R, what is this one gonna look like?
Capital Y, capital R.
In the exact same spot. Does that make sense? If you do the top one, again going top down, whatever the first one is, you just carry it all the way through. You don't have to think about it, okay? You just gotta think about the first one. What about this one? Where does the capital Y go? First spot or second slot? First. What about the capital or lowercase r?
the second slot.
Second slot, just like that. Now, for each of these, what do? I go capital Y, capital Y, capital Y, lowercase r, lowercase r, lowercase r, just like that. Again, all the way down. Does that make sense, what I'm doing? Okay. Just being a little bit more methodical about it rather than just doing it all willy-nilly, okay? Next one, lowercase y, where do I put it? First or second slot?
Second.
Second. Capital R? First. And what do? Just
Bring it
bring it all the way down. Just like that. Then over here, I have lowercase y, lowercase r. They're both gonna go in the second slot. If you notice right here, you can also draw a line underneath the capital letters, just so that you know that they're the capital letters, just in case, okay? Now that I've done the top down, now what can I do? The left. So if you notice, the only other spot that this Y can go is what? The second slot. And the only other place that the R can go is what? The second spot. Capital Y, second spot. What about this capital R? First spot. Again, you see why we do the top down, because when we go across, there's only one spot for it, so we really have to think about it. We just fill in the blank. Does that make sense? Okay. Again, capital Y, lowercase r. Capital Y, lowercase r. Capital Y, lowercase r. Just like that. Now I have lowercase y and capital what? R, lowercase y, capital R, lowercase y, capital R, lowercase y, capital R, lowercase y, lowercase r. All the way through. Now what we're gonna do is, we're going to try to figure out what exactly, how many we have of each of these. So this one is yellow, or excuse me, green and round, green and wrinkled. Yellow and round, yellow and wrinkled. I want to know how many yellow and round I have from my dihybrid cross that I just did. So I'm gonna go through each person, and I want you guys to tell me if they are yellow and round or whatever we're looking for, okay? So, for example, this first one, is it yellow and round? Because we're looking for yellow and round first, okay? Is this yellow and round? Just say yes or no. Isabel, this first one. Is it yellow and round? Yes or no? What does this capital mean? That it's, this capital R means that it's, so it's yellow and round. So since it's yellow and round, that's one. Sammy, is this yellow and round? Yes or no?
Yes.
Is this yellow and round, Matthew? Is this yellow and round, Emily?
Yes.
Is this yellow and round, Jonathan? Is this yellow and round, Abigail?
No, it's yellow.
You don't have to tell me, just say yes or no. No, you're fine. Is this yellow and round, Abraham?
Yes.
Is this yellow and round, Savannah?
No.
Is this yellow and round, Christian?
Yes.
Is this yellow and round, Nicholas?
Yes.
Is this yellow and round, Itzel?
Oh, no.
Is this yellow and round, Itzel? Or sorry, Sarai?
No.
Say it again.
No.
Is this yellow and round, Isabella?
Yes.
Is this yellow and round, Sammy?
No.
Is this yellow and round, Matthew?
No.
Is this yellow and round, Emily? Say it again.
No.
So now, since we've done all of them, how many of these are yellow and round? 1, 2, 3, 4, 5, 6, 7, 8, and 9. Just like that. Okay, now we're looking for yellow or green and what?
Green.
Green and round. So, same thing. Is this green and round? Yes or no? Jonathan?
Yes.
What is it?
Green and red. So no.
It's yellow and wrinkled. We're looking for green and what?
Round.
Round. So, is this green and round, Abigail?
No.
No? Or no? If it's not green and round, then what is it?
Yellow and, wrinkled.
Perfect. So, is this yellow and, or excuse me, green and round, Abraham? Yes. So, this is 1. Is this green and round, Savannah? What does this mean right here? Is it yellow or green? These are lower cases.
Green.
What does this mean? That it's round or wrinkled? So is it green and round? Yes. Is this green and round, Christian?
No.
Is this green and round, Nicholas?
Yes.
And then the last one is not green and round. So again, how many is this that we have? 3. Next one, what we're looking for yellow and what? Wrinkled. So is this yellow and wrinkled, Itzel?
Yes.
Yes. Is this yellow and wrinkled, Sarai?
Yeah.
Is this yellow and wrinkled, Isabella?
Yes.
Is this yellow and wrinkled, Sammy?
No. Wait. Yes.
This is green and wrinkled, so it's not yellow. But that's how many? 3. And since this is the last one, this one is again green and wrinkled, so there's only 1. So again, also you notice that when we go to count, another thing that we can do to make it easier for us is you can use different colors, or you can use different shapes or things to kind of cross it out, just to make it a little bit easier for you to count. Again, when you go through it, then you can identify which one you messed up on, especially if you feel like you messed it up. Okay? Does that make sense? Okay. So just a couple of more things before we wrap up the last bit. So again, we kind of talked about these words over here, but I want to make sure that we understand the difference between principle of segregation and principle of independent assortment. When we look at principle of segregation, what exactly is it? Go ahead and read it for me, please. Matthew.
Factors known as alleles on chromosomes are separated from each other in meiosis. A1 to and you have an equal count of inheritance.
So again, this means principle of segregation. Remember that during meiosis, specifically anaphase 1 and 2, remember the chromosomes are separated, and when they separate, they go into 2 or 4 what? New haploid cells. So those haploid cells become gametes, which are egg and. But when they develop into egg and sperm, you have an equal chance of inheriting any one of those alleles. That's all that means. One more time. Principle of segregation says that when those chromosomes separate from each other during meiosis anaphase 1 and 2, that they do so randomly, and you have an equal chance of what? Inheriting. You don't have a higher chance of inheriting the dominant one or the recessive one. It's just what you get. Okay, it's all random. While principle of independent assortment means what? Go ahead and read it for me, please, Emily.
Alleles of a trait are
Inherited.
inherited independently of each other.
What that means is, when we look at these as examples, you can be yellow and what? Round, or you can be yellow and you can be green and round, and you can also be green and what? That means the principle of independent assortment just means that two specific traits are inherited independently of each other. That just because you're green doesn't mean you have to be wrinkled. Just because you're green doesn't mean you have to be round. Just because you're yellow doesn't mean you have to be what? Or just because you're yellow doesn't mean you have to be wrinkled. Again, traits are inherited independently of one another. But I want to clarify that these traits are all what we call Mendelian traits or Mendelian genetics, where things are very straightforward, they're very cookie cutter. It is what it is. Okay? When things start getting muffled or kind of like gray, that's what we call non-Mendelian genetics. Okay? Because it's not so cut and dry anymore. Okay? Any questions? Okay. So the last thing I want us to go ahead and do is to go ahead and look at questions 22 and 23 on the back of page 12.6. 22 and 23 on the back of page 12.6. So 12.6, 12.6, 22 and 23, which are on the right side. So just for clarification, I know the numbers are all messed up because I can't count, as you guys can see. So 22 and 23 on this side, these two are the ones I want you guys to do, okay? They're just dihybrid crosses.
Mr. Stewart,
Yes.
is our test next week?
Yes, next Thursday. That's what the reminder said.
Yeah, that's what I was asking, just
Yeah.
making sure.
Our exam 4 is next Thursday?
Mm-hmm. Yeah. Oh, by the way, what we just did, the dihybrid cross, that is worth 15 points on exam 4. Okay.
Wait, so what can I use ours?
Now I have to actually.
We only have 5 minutes left.
Wait, did you say you're gonna check journal?
I'm gonna do it on Friday.
Alrighty.
Abraham and Matthew, are y'all going on the trip? Oh, Jonathan, you're going on the trip? So if you're going on the trip, I need to do it Thursday, but if you're not, then
Oh.
you